Good evening, everyone, and welcome to Flanders Vaccine's third COVID-19 webinar. The first and second session, which took place in June 2020, focused on preclinical and clinical research for the development of a vaccine candidate for corona and on the potential of some drugs for the treatment of COVID-19. Today, I'm proud to welcome you to a webinar discussing the already approved corona vaccines in Europe. And this only 9 months later, which is a real success. However, Europe is still experiencing significant challenges due to this pandemic, but yet there is hope, and thanks to the prioritized and safe development of several COVID-19 vaccines and the huge efforts of researchers, industry, governmental organizations, and healthcare providers, and so many other people, the vaccination campaigns could be rolled out in several countries. But this is also accompanied by many questions and uncertainties among the people. Are these vaccines safe? Do they affect fertility? Are these different types of vaccine protective against current and upcoming COVID-19 variants? With this webinar, we want to inform you better about the different types of COVID-19 vaccines available and to provide some answers to your questions. Before I give the word to the moderators of this evening, Professor Piet van Damme, vaccinologist at the University of Antwerp and Vice Chair of Flanders Vaccine, and Professor Isabel Leroux-Roels, also a vaccination specialist at the University of Ghent and the University Hospital of Ghent, I really want to thank all the experts that contributed to this webinar, the speakers, panelists, and of course also the moderators. Thank you for your time, especially after working hours, and to share your expertise with us. I wish all the attendees a very inspiring evening, and I now pass the word to Professor Isabel Leroux-Roels.
Yes, good evening, everybody, and welcome at this scientific webinar on COVID-19 vaccines. Before giving you an overview of the very interesting program we have put together for this evening, I would like to inform you of a change of moderators. I was going to be accompanied by Professor Piet van Damme this evening, but because of his obligations in the task force, he was unable or is unable to join us. And luckily, I found somebody who was available and willing to join me here in Antwerp. I would like to introduce Professor Geert Leroux-Roels as my co-moderator. He is the founder of the Center for Vaccinology. He's an expert in vaccinology, emeritus professor at the Ghent University. So now I would like to give a brief overview of the presentations of this evening. We will start with an overview talk given by Professor Jean-Michel Dogné on the efficacy and safety of COVID-19 vaccines marketed in Europe, followed by a presentation on the epidemiology and genomic characterization of COVID-19 virus variants by Professor Piet Maes. And then the third talk of this evening will be on perspectives for next generation COVID-19 vaccines by Dr. Marco Cavaleri. The three presentations will be followed by a panel debate, a Q&A session with our three speakers, and we will also welcome two additional panelists, panel members, Dr. Johan van Hoof, Global Head of Infectious Diseases and Vaccines from Janssen Vaccines, Johnson & Johnson, and Dr. Stefan Lockhart. He is the Vice President and Head of Vaccine Clinical R&D Europe and Asia Pacific, at Pfizer. There will be no questions in between the talks, so all questions will be asked. And I would like to thank you already for all the questions you already sent in prior to this webinar. Some best practices before I give the floor to the first speaker. So it is important that you know that you are all in listening only mode, so no need to mute. You are muted already. There is no chat function, but questions can be asked within the Q&A section. So there is a button in the upper right corner of your screen. So please, we have already received many questions, but please feel free to ask additional questions. As I already told told you, we will try to answer as many questions as possible during the panel debate, but I can already tell you that it will be impossible to answer all, but we will, of course, do our best. And then last, important to say for the Belgian physicians who wish to acquire accreditation points at the end of the session, at the very end, there will be a link that will be posted in, it will be present on the slide and posted in the Q&A section, and you need to click on that link in order to complete your details, your the required details in order to get the accreditation points. And this link is only accessible until midnight this evening. Of course, the evaluation form will remain open for a longer period of time, and we of course always welcome your evaluation. Important also to thank our strategic partners. So Flanders Vaccine is sponsored by the 6 strategic partners which are shown here on the slide. So that brings me to introducing the first speaker, Professor Jean-Michel Dogné. He's a full professor at the University of Namur. He's an expert in pharmacovigilance and Belgian member of the European Medicines Agency. He is also a member of the Global Advisory Committee on Vaccine Safety at WHO. And as I said, he will be talking or giving an overview of the marketed COVID-19 vaccines in Europe. Jean-Michel, the virtual floor is yours.
Thank you very much. So I hope you can see my slides.
Yes.
Yes, okay, so thank you. I will go on. So I was asked to discuss about the characteristics of the authorized COVID-19 vaccine and some identified and potential risks. And of course, it's a major, I would say, issue now, since there are many vaccines that have been authorized. The latest have been authorized today, so I updated my presentation today. And I also want to say that I'm a member of the Federal Agency of Health and Medicinal Products at Belgium level, and member of the PAC of the European Medicine Agency and the WHO, the Global Advisory Committee of Vaccine Safety. But this is, in this presentation, my point of view and not the institution that where I'm sitting in. And I do not have any conflict of interest as being part of this institution. So I guess now that you well know the basics of the vaccine development and the fact that we are mainly working on the spike protein on the virus being the one that leads to the entry within into the cells via the ACE2 receptors, and the immune response is therefore a trigger via the spike protein as well in antigen-presenting cells, that leads to T helper cells, which lead to B cells, and therefore humoral response with antibody response, but also cellular response. And for these two, we have also long-lived memory B and T cells. And so basically, the vaccines that are currently under development are classical vaccines, such as inactivated virus vaccine. This is mainly the approach taken by some Chinese companies that have developed the inactivated virus vaccine. And if you go now to more specific approach, we have some vaccine under development, of which Novavax is currently on the table of the European Medicine Agency in rolling review. This is a protein subunit, which is adjuvanted as well. And we have, of course, two major vaccines now with the AstraZeneca, and from today on, the Janssen vaccine, which are non-replicating viral vector, which are adenovirus. There are some difference in between the two companies. Shadox is a chimpanzee adenovirus vaccine, and the Johnson & Johnson is a more, I would say, classical adenovirus vaccine. Of course, the development of some specific vaccine were, I would say, not expected as rapidly as the RNA and DNA vaccine. The RNA vaccine won the race, of course, because now we have at least 2 vaccines in marketed, at least not only at EU level, but also worldwide. And we have also other RNA vaccines currently discussed also at the level of the European Medicine Agency. And I will just talk about on that. For the one who do not know or are familiar with the principle of the vaccine development in EU level, I just want to remind that there are 3 major stakeholders. The first stakeholders being the vaccine developers, so the applicant at the stage of the marketing authorization, who propose a review of the data at the level of the European Medicine Agency. What is currently now assessed at the level of the European Medicine Agency is a rolling review of all the data from the different vaccines. And the European Medicine Agency mainly works with a series of committees, including the CHMP and the PRAC. And I'm sitting in the PRAC for Belgium. And there is, on top of that, I would say, EMEA Pandemic Task Force, which has a series of members from different committees that also take into account the different data discussed at the level of the PRAC and the level of the CHMP. At the end of the day, it's the CHMP that proposed a recommendation on the benefit risk of the vaccine, and this recommendation is sent to the European Commission. And it is formally the European Commission that provides the marketing authorization. In the case of the vaccine, since the majority of the studies are still ongoing and there are additional information sometimes needed, this is a conditional marketing authorization. And based on this conditional marketing authorization, all the 27 member states at EU level have the possibility to have the marketing authorization. On top of that, I will not discuss the fact that the European Commission for a series of the vaccine has also proposed to work with the pharmaceutical industry and vaccine developer for having some specific vaccine and some specific doses at different member states, meaning some specific contracts. And this was usually done before the marketing authorization. And just to go rapidly now to the core of the presentation, we have the vaccine landscape at EU level, and you see that on the 4 first vaccine, Comirnaty, Moderna, AstraZeneca, and Janssen, they have been submitted for rolling review early in 2020 for the majority of them. The EMEA opinion has been proposed for the first one, Comirnaty, at the end of 2020, 21st of December, early January for the Moderna, AstraZeneca at the end of January, and today for the Johnson Janssen vaccine. And the quantity of vaccine has been associated to a contract with the European Commission, usually based on the demographic of Belgium in EU level, which represents around 2.5% of the population. And importantly, you see that the same day, the European Commission proposed a conditional marketing authorization of the European Medicine Agency opinion. Of course, they are different, as I have said, and we will see that. We have the 2 mRNA vaccine for Pfizer and Moderna, then the 2 adenovirus vaccine, which are a bit different in between AstraZeneca and Johnson. And the other 2, and there will be an additional one, are also in rolling review since February of 2021, the CureVac vaccine, which is another mRNA vaccine, and Novavax, which is a protein subunit in nanoparticle adjuvanted. The majority are in 2 doses, except Johnson & Johnson, but we will see that it may also have some impact regarding the data, the efficacy, and the possibility to have a second dose in subsequent studies. So I think that you are now well aware of what was published with the 2 mRNA vaccines, so I will not go through too much in details of the efficacy and the safety of what was published at the time of the marketing authorization in New England Journal of Medicine. But you know that for the Pfizer vaccine, what was quite astonishing is that for the first time where we had important clinical trials with mRNA vaccine, it led to quite rapid and efficient development on a very efficacious vaccine with 95% of efficacy in studies over than 30,000 people. And you see that not only on symptomatic COVID-19 vaccine, but also on severe COVID-19 vaccine, we had a quite impressive efficacy. And for this case, this was mainly assessed 7 days after the second dose. And I remind you that the second dose is to be used after 21 days. And this, I think, is quite important also to understand that after the first dose, we start to have an efficacy, and the efficacy starts usually around 12 days. And I would say basically this is from 12 days after that you see really an efficacy that increases. But it means that two things. After the first shot, of course, for a certain period of time, you may still be infected because you are not protected. And we have heard in the media that indeed some people are still infected after the first dose before the second dose, and mainly before 2 weeks. And of course, this may explain, of course, the situation. But quite interestingly, if you take the efficacy not from 0 to 21 days, but from, I would say, 14 to 21 days, then the efficacy starts really to be high and nearly as higher as 1 week after the second dose, so nearly 90%. So that's quite important to be very careful before the second dose, but also after the first dose, 2 weeks after the first dose, you start up to the second dose to have a high efficacy, and I will show you that in real-world data studies. What was quite impressive, and this is always what we have some doubts about the efficacy of the vaccine, when you go with age, you may decrease the efficacy of the vaccine because of immunosenescence. And in this case, this was not the case, since over 55 at 65 years of age and even 75 years of age, the efficacy was maintained. And this is quite important. Also, the number, of course, the figures are lower and the variability is higher, but this is quite consistent with the important efficacy of whatever the group of age. And this is also the case for the comorbidities. I do not propose you the slide in this case, but especially for the comorbidities, the more frequent comorbidities being usually obesity. And what is the proof of concept of the mRNA vaccine was that two vaccines developed in parallel, I would say, provided the same estimates in terms of efficacy, and the second one being indeed Moderna. And if you look at the efficacy data in clinical trials, with the same setting, I would say, of the clinical trial, you see also that you reach the same level of efficacy, around 95% with the Moderna vaccine. And quite importantly, you see a 100% efficacy on the severe COVID-19 cases. So this was also quite impressive, but quite interestingly, also led to a validation of the proof of concept that the mRNA vaccine targeting the spike protein was really a good target and a good vaccine. There are some differences, of course. This is in a modified intention to treat analysis, the cumulative event rate and the days since randomization. This is the same, I would say, curve that we have with the Pfizer vaccine. There are some differences. The Moderna vaccine is administered after 28 days, and the assessment of the data was proposed 2 weeks after the second dose, which is administered after the first dose, 28 days after the first dose. Again, you see that you start to have difference in terms of efficacy after 2 weeks, and from 2 weeks to the second dose, you increase indeed the efficacy. So that's quite also important that you see that we have the same level of protection after 2 weeks with the Moderna vaccine. And the estimates about the efficacy over 65 years of age was still good. It seems that it may be a bit less important than what we have with the Pfizer vaccine, but we need to be very careful here because the numbers are quite limited, are quite small, and the variability is quite high, and the studies are different. So it's they were not head-to-head comparison studies, so we cannot say that one of the two vaccines is really better over 65 years of age. What is important is that we keep important efficacy in this group of age. The same being true for the comorbidities. Now, when it comes to AstraZeneca, which was marketed later, it is also a 2-dose, and we will come to that, and we will come to the difficulty to understand really how we need to interpret how and when the second dose need or may be used. And I would say the clinical trial program was a bit different and was not such straightforward that we had with the Pfizer and the Moderna vaccine. So it is also a genetically modified organism, since it's adenovirus that contains DNA that will then be changed into mRNA and then put in protein S in the cells and then induce the immune response. As I have said, the clinical trial was a bit more not complicated, but a bit more, I would say, dispatched in between countries and in between setups, and there were different setups, and you have certainly seen that in the literature, that we have not only different countries, but in the different countries we might have some different characteristics in terms of participants. For instance, the first one was up to 55 years of age, and you see that we had some different dosage that were proposed, standard dose and low dose. And in published data, we have seen that in some cases, with a lower dose first and second dose being higher, we might have a higher activity. But still, at the end of the day, in accordance with the company and the European Medicine Agency, the assessment was mainly based on the two main studies, the COV-02 and the COV-03 in UK and Brazil. And this is the standard dose that is used, first and second dose, and there are also some difference in between this vaccine and the other two vaccines, is that in some cases, the comparators was another vaccine, a tetravalent meningococ. And you see that for the administration of the second dose, it's not such clear cut with what we have with the Pfizer and the Moderna vaccine, which are respectively 21 and 28 days. We could go up to 12 weeks for different reasons. I will not go into detail, and mainly for, I would say, catch up of a second dose when initially it was mainly proposed a first dose. So what does it mean? It means that the data may be interpreted differently based on different variables. And what needs to be taken into account, as we had with the Moderna vaccine, is that the second dose, the assessment of the efficacy was usually done after 2 weeks after the second dose, but you see that the interval may be 4 to 12 weeks. And if we take the overall data about efficacy, we have indeed, with the second dose being 4 to 12 weeks, an efficacy that is lower when compared to the mRNA vaccine, around 60% of efficacy. But still, what was quite interesting with this vaccine is that if you take at least one dose for the first dose and 22 days post-dose one, so at least 3 weeks post-dose one, you see that in all participants who receive at least one dose after 22 days, you see that you do not have any cases of hospitalization. So an estimation of 100% of efficacy on hospitalization was proposed by the European Medicine Agency in the European Public Assessment Report. And indeed, the European Public Assessment Report was published quite recently as well on the website of the EMA and provide more details about some specificity regarding the second dose. And you see that if you pool the data coming from the two main studies and the second dose that may be administered 4 to 8 weeks, 8 to 12 weeks, or more than 12 weeks, we might have efficacy that may be quite different. In fact, if you go with 8 to 12 weeks, which is recommended by the WHO, and 12 weeks and over, you have an efficacy that increased from 60 to 75%. So the efficacy seems indeed dose dependent, especially for the second dose. So the question is, yes, but what does it happen after the first dose of AstraZeneca? Do we have to wait up to the second dose to have an efficacy? I have shown you that with Pfizer already 2 weeks after a first dose, and with Moderna 2 weeks after first dose up to the second dose, you can have an efficacy that is already quite high. And in this case, if you take indeed the post-dose one just to the second dose, you have an efficacy that may seem quite limited, up to 42.8%. But again, if you start after 3 weeks after the dose one, which is the timing that needs to be mainly focused on, you do not have hospitalization, I have shown you after that. But if you take the efficacy in between the first dose and the second dose, 21 days after the first dose, you have a 73% of efficacy. So you do not have to wait up to the second dose to have such a level of efficacy. And the same is also true if you use the second dose after 22 days, the efficacy 22 days after the dose 1 up to the week 8, week 10, or week 12 for the second dose, you have efficacy that starts to reach up to 80%. So that's quite important to see and to understand the differences that we have with this vaccine when compared to other vaccines. And some data were also published on the Lancet, and I must say that it may be a bit confusing for some of you who read the data because in terms of efficacy, of course, every day you may have efficacy that may, I would say, change lightly depending on when the interim analyses are performed, but these are the data from what were submitted at the level of the European Medicine Agency. I can say that in the Lancet, these are not exactly the same estimates, but we are in the same, I would say, range of estimates after the first dose, before the second dose, and after the second dose, depending on the timing of the second dose. When compared to Pfizer and Moderna, you know that there were some limitations, not in terms of efficacy, but in terms of estimation of efficacy, especially over 55 years of age. And the EMA was quite clear that the indication was above 18 years of age, and Pfizer, just to compare, is above 16 years of age, and Moderna is above 18 years of age, but they did not provide any age limit. Also, they had some difficulties to provide figures in all the participants over 55 years of age, not because the vaccine does not work, but because the level, the power to provide some figures is quite limited. And so they agreed to say that the vaccine could be used over 55 years of age, given that we have an immune response that is provided, especially in terms of antibodies and neutralizing antibodies, and with the experience that we have with the other vaccine. But you know that in some countries, in some member states at EU level, they limited the use at 55 years, up to 55 years of age, or for instance, up to 65 years of age. And you know that Belgium estimated that we should recommend the use up to 55 years of age, and now they have proposed to use it over 18 without age limit, and I will show you why. Why? Because I think for three vaccines, it is important that we now rely also on the real-world evidence of the effectiveness of this COVID-19 vaccine. And there were a series of now data that have been preprint or that have been published, and I will present you some of them, and I think this is quite important to see what's happening in real life other than clinical trials. And the more robust in terms of evidence base is the one that has been published in the New England Journal of Medicine quite recently, which is a match-controlled study in the data from Clalic Health Services with the Pfizer vaccine in the Israeli cohort. And it's quite important because you will see that they propose an observational data, an observational study, which estimates the vaccine effectiveness in 5 outcomes of interest. And the 5 outcomes of interest are symptomatic illness, hospitalization, fever, disease, and death. And quite importantly, you see that this is the days after the first dose and 7 days after the second dose, so per protocol in the clinical trials when we estimate the efficacy 7 days after the second dose. And you see that basically after the first dose, you have a 57% efficacy in between 14 and 20 days after the first dose, and it increases in between 21 and 27 days after the first dose. So it means that after the first dose, we have already an important efficacy on symptomatic illness, on hospitalization as well, and this is quite important to have this data, of course, in real life, and on severe disease and even of death. But quite importantly, we see also, and we are under discussion and under pressure to see, do we use only one dose, or do we delay a second dose, or do we even not use a second dose? And you see that in this case, 7 days after the second dose, for all estimates, for symptomatic illness, hospitalization, severe disease, except for death, for which we do not have the data, the efficacy overall increase, whatever the endpoint. Symptomatic illness up to 94%. So you see that we reach the exact level of estimate that we have in clinical trials, and 92% in severe disease. And so this study really confirmed the clinical trial finding. It demonstrates the booster effect, so the importance of the second dose, and that the effectiveness increases as time passes, especially after the first dose, before the second dose. And the only, I would say, limitation that we have is that if we had some age group, and more specifically, if we have comorbidities, we might have a slightly lower effectiveness, but the efficacy was still higher in elderly population. The second paper that was not published yet and that was not yet peer-reviewed, but which is quite also important, is the one that deals with the data in Scotland after a single dose on hospital administration. And why is it important? Because it provides information not only for the Pfizer vaccine, but also for the AstraZeneca vaccine. And why is it also important? Because you know that we have limited in Belgium at first sight, I would say, up to 55 years of age, the AstraZeneca vaccine. And you see that in the UK, the older you get, the higher chance you have to have the AstraZeneca vaccine, which are in red. And even after 75 years of age, you mainly have an AstraZeneca vaccine. So it will provide information for the AstraZeneca vaccine, but also in the elderly population. And what did this study provide us is that indeed for both vaccines, they peak after a single dose, after a single shot, an efficacy which is higher after 28 days, and especially in between 28 and 34 days after the first dose. For the Pfizer vaccine, it was 85%, and for the Shadox vaccine, so the AstraZeneca vaccine, it was 94%. But one of the most interesting data that needs again to be assessed more in detail and peer-reviewed and published is that after a certain the estimation of the vaccine efficacy after a certain age, over 80 years of old of age, where we have mainly the AstraZeneca vaccine, the efficacy is still 81%. So it is quite important to see that it may drive us to understand that AstraZeneca was also quite efficacious in over 65 years of age. And finally, as a third study that I want to propose to you in real life that needs really to be assessed carefully, because I think there are many limitations of this study that needs to be taken into account. Just it is for information now. It's also the vaccine efficacy against symptomatic disease and hospitalization and mortality in all the adults in England. And again, it is a study that is a test negative case control design in this case, which is usually used for the flu vaccine to assess the effectiveness in real life. And it deals also with the two vaccines, Pfizer and the Shadox vaccine, so the AstraZeneca vaccine. Again, there are many limitations in these studies that will need to be discussed by the reviewer and where it will be submitted. But in over the 70 years of age, it provides quite important information after first dose that the Pfizer vaccine has indeed an efficacy or effectiveness over 60%, and then it plateaus. This is represented in this case. And this also highlights the fact, and this in the study they have also shown, that if you have the booster dose, so the second dose, mainly after 21 days, you have an efficacy that increases as we see in clinical trials, and if we have seen that we have seen in the New England Journal of Medicine. For the Shadox vaccine, we have seen a 60% efficacy after the first dose, starting from mainly the main activity 28 days, and this is again quite important since we know that we delay the second dose up to 8 to 12 weeks. But there are some, I would say, important limitation in these studies that need to be taken into account. For instance, if you take the Shadox vaccine, the odd ratios when compared to the adjusted odd ratio, we might have strong differences. So it means that the adjustments there lead to some huge modification, and there is a need to see what are the reasons for these adjustments. It provides also some information of the vaccine efficacy against hospital admission, within 40 days of testing positive. It means that if you are tested positive, what is the chance that you have to have an hospitalization if you are vaccinated or not? And you see that it is approximately 40% for both of the vaccine, for the Pfizer vaccine and for the AstraZeneca vaccine. And if you take the vaccine efficacy against mortality within 21 days of testing positive, this is 51% for the Pfizer vaccine. So again, this needs to be taken with a lot of caution based on the, as usual, based on the limitation of these kind of studies, but all point to the same direction, that these vaccines prevent severe disease, hospitalization, and death, whatever the vaccine up to now. So these are the limitations. And finally, the discussion of today, and if the meeting was yesterday, I was asked to present the vaccine that have been marketed at EU level. I would not have presented the Johnson & Johnson vaccine. Also, we had data, of course, from the FDA that has marketed the Johnson & Johnson vaccine earlier than at the level of the European Medicine Agency. Again, a bit different vaccine, but we have some information, interesting information with this vaccine, especially, for instance, for pregnant and lactating women, because this is an adeno-26 viral vaccine, and this is the same platform that is used for the Adeno vaccine, which is marketed now, and for which we have an experience in real life, especially, for instance, in pregnant women. And this is also a replicant deficient vaccine, and this will be, in this case, a single dose for which we have the data presented by Johnson & Johnson, and quite importantly as well, as it is for AstraZeneca, the storage can be done in between 2 and 8 degrees, which was not the case, of course, for the 2 mRNA vaccines. And the clinical development plans was also quite huge, and we have 2 ongoing studies, 2 main ongoing studies. The one that led to the marketing authorization, which is called the COF 301, which is a phase 3 study that studies a single dose with over 40,000 people in 8 countries, and it will have an interest to have 8 countries. But I told you that there might also be an interest to have a second dose, and there is an ongoing study with a 2 dose of the adeno vaccine. So the question was a single dose interesting to have a marketing authorization of this vaccine based on this important study for which we have a higher power than for other studies with, for instance, some mRNA vaccine and for the Shadox vaccine. And they studied the efficacy mainly after 14 days post-vaccination, dose 1, because there's only a single shot, and 28 days post-vaccination. And quite interestingly, again, is that in this study, and it's quite important, elderly population, meaning over 65 years of age, represent 20% of the population, but 20% of high-powered study. So it means that we have many cases, and secondly, in comorbidities, we have around 40% of people with comorbidities. So it was really a study that was quite important in terms of estimate about the efficacy. And you see that regarding the efficacy, 14 days post-vaccination in all subjects, we have a vaccine efficacy around 67%, and which was kept in elderly population. And even more, you see that after 14 days in elderly population, also the estimates are based on a low number of cases, but it increased with age, with 82 and even 100%. No case over 75 years of age after 40 days post-vaccination were detected. So the vaccine efficacy was quite important already after 40 days, and it was maintained after 28 days post-vaccination. Again, over 65 years of age, it is maintained also after 28 days, but the limit with more limited cases, of course, which undermine the estimates in terms of variability around the estimates. Quite importantly, again, the severe cases, and you see, and this is again something very interesting, is that 28 days post-vaccination, the severe cases, it's the efficacy on the severe cases is quite huge, up to 85.4%, and that's quite important as well. And if you take after 14 days, the severe cases, you have 14 with the Janssen vaccine and 60 with the placebo, and that's quite important. And you see that if you look now at the hospitalization in this group, you had 2 versus 6 that were hospitalized, so basically somewhere that as an efficacy on hospitalization up to 75%, and 3 individuals died, all in the placebo group. So again, very interesting result with a single shot of the Janssen vaccine, but this is also the one that provided very interesting data on in region where we had some variants. And if we take the three main countries where we might compare, I would say, the results, and we have US, Brazil, and South Africa, and we knew that, and this was really interesting from the company, that they made a laboratory confirmed analysis of what was the variant of concern in the population. You see that in the US, it was mainly the Wuhan H1 strain, so the original strain. In South Africa, it was mainly the South African variant that will be discussed after, and in Brazil, it was mainly the Brazilian variant. And you see that, interestingly, especially the point estimate on severe cases, that you especially, let's take 28 days, you had 85% on efficacy on the US database, and you have still, after 28 days, in the Brazilian database, the same level of efficacy on the Brazilian variant mainly, and a short, I would say, decrease about the efficacy in the South Africa. So it's quite important to see that for the one who fears the efficacy of the vaccine on the variants, when we look at the clinical estimate, the clinical endpoint, it may be different about some estimate that we might have with pseudo-neutralization testing in laboratory. And I will now finish by, I would say, some safety data. You know that that's some difficulties that we have had today and for 3 days at the level of the track, but the safety data, interestingly, came first from the clinical trials that were quite well powered to identify cases which are rare cases, which is usually not the case when we have clinical trials with 3,000 participants. In this case, it was usually more than 30,000 participants. And I would say that the reactogenicity is what is expected with the vaccine, and we might have some differences. For instance, for the Pfizer vaccine, the Comirnaty vaccine, we have higher frequency of pyrexia was observed after the second dose, and mainly in younger individuals, less than 55 years of age. And it's quite important, you will see with the AstraZeneca vaccine, we know that we have flu-like syndromes that mainly occurs after the first dose when compared to the second dose. So the profile of adverse events may be a bit different. And you see in this case that we have small imbalance for acute peripheral facial paralysis with the Pfizer vaccine that was able to be seen in the clinical trials. The anaphylaxis were not seen in clinical trials, but mainly post-marketing, and this is why they were mentioned in the SMPC of the product. And I would say this is the same for, exactly the same for the Moderna vaccine, some cases of acute peripheral facial paralysis, otherwise I would say classical reactogenicity, and we have seen anaphylaxis mainly post-marketing. I will come back to that. As I have said, with AstraZeneca, I think the overall safety profile is the one that would be expected with a vaccine, except that we have now more reported flu-like syndrome, and mainly after the first dose. And indeed, this is what we see, basically, that the profile is maybe a bit different in between AstraZeneca and mRNA vaccine, especially flu-like syndrome after the first dose. And for the Janssen vaccine, this was published today on the website of the EMA, we see that we have also quite classical reactogenicity associated. In this case, of course, we have a single shot and not two doses, so we cannot compare the first and the second dose. What we can see in this case is that anaphylaxis has been added as well in the SMPC of the product, because there have been 1 cases in clinical trials that were developed lately, and so it is also proposed in the SMPC. But again, what is quite important is now the post-marketing assessment of the safety cases, the adverse event, and what I can tell you is that at the level of the Belgium, but also at EU level, we have a large amount of cases reported, just spontaneous cases, but I would say these are more stimulated cases because we simulated different member states to report as much as possible cases. And indeed, what you see is that we received many cases up to the 11th of March 2021. And this is quite huge when we know how many cases usually receive Belgium a year for all medicine, which is around 1,000. In this case, in less than 3 months, we received 6,000 cases only for the 3 vaccines. And you see that for the AstraZeneca vaccine, we have proportionally maybe more cases that we have with the Comirnaty vaccine, mainly associated to non-serious cases of flu-like syndrome. And these flu-like syndrome were also identified at the level of the French national authorities, and they proposed to have a staggered approach, not to vaccinate all the people from the same department, for instance, at the same time. And they also proposed to use paracetamol. And quite interestingly, in the AstraZeneca vaccine, when you see the different studies that we have assessed for the safety, the fourth study, you see that in prophylactic treatment, paracetamol was proposed. This is the case also for the other vaccines, but for instance, in the COVID-3 vaccine, in the Brazil study, the paracetamol was systematically used. So in these clinical trials, of course, it may underestimate the flu-like syndrome. So this is a recommendation that was proposed by France to use paracetamol systematically to prevent or to treat the flu-like syndrome cases. When it comes to the anaphylaxis, this is really the real signal that we had first post-marketing with BioNTech and Moderna vaccine. Why? Because they were not seen in clinical trials and they were added as an important identified risk post-marketing and in the SMPC of the vaccine. And I remind you that this is something that is quite well known with the vaccine. The rate of anaphylaxis is usually in adults and infants 1.3 per million vaccine dose, whatever the vaccine, I would say. And in the CDC assessment of the cases post-marketing for Pfizer and Moderna vaccine, they have it estimated 4.7 million doses for the Pfizer vaccine and 2.5 million cases for the Moderna vaccine. What is interesting to know is that it mainly appears before 15 minutes, and secondly, it mainly appears in women, especially for the mRNA vaccine, but also for the AstraZeneca vaccine. And this is quite important because we have made some recommendation how to minimize this risk as much as possible, for instance, a follow-up of 15 minutes after the vaccination. And at that time, it was not, I would say, labeled anaphylaxis for the AstraZeneca Oxford vaccine. This was discussed this week at the level of the PAC, but it was also already discussed earlier this month at the level of the MHRA, so in UK. And you have seen that in UK, when you compare the number of cases, 190 cases of spontaneous report of anaphylactoid, I would say, reaction out 9.5 million as denominator of Pfizer vaccine, we have the same kind of level of estimate on spontaneous reporting reported with the AstraZeneca vaccine. So they've updated the SMPC based on the risk of anaphylaxis, which is in line with what the company supported also at the level of the European Medicine Agency. So now all the vaccines are equal in terms of level, I would say, of identify risk in terms of anaphylaxis, and what is the more important is to follow for 15 minutes individuals treated with the vaccine, whatever the vaccine, and have the supervision and appropriate medical treatment. Every month we do assessments of all the data that come from the company and spontaneous reporting. We published at the level of the European Medicine Agency monthly report. For instance, on the 4th of March 2021, we have identified diarrhea and vomiting as frequently reported and updated adequately the SMPC. And we do signal detection quite frequently in the European database for different events of interest. And what we do usually is observe versus expected analysis. So we need to have the background incidence rate that we have had in the past at the level of the European Medicine Agency as part of the program and access program that allow to have these background incidence rate. And this is now quite important because we received a huge level of reports from the different countries, not only at EU level, but outside EU level, mainly driven by the patient exposure, of course, which was first for Comirnaty, then for Moderna, for which the patient exposure at EU level is a bit lower, and AstraZeneca, that also take into account cases from the MHRA. And this week, for instance, at the level of the PAC, published on the agenda of the PAC, you might have seen that we have discussed thromboembolic events based on the two cases of first initially coming from Austria, and now the cases coming from different member states, including Denmark. We have discussed immune thrombocytopenia for the 3 vaccine. We have discussed for Comirnaty localized swelling in person with history of dermal filler injection and anaphylaxis for AstraZeneca. So this was only for this week at the level of the tract. And you know that it went to the media, especially for the suspension of a batch in Australia with 2 cases, and that we notify at the level of the EMEA and the tract that the link between the batch was quite difficult to identify, and that we will study, of course, the cases of thrombotic events associated with the vaccine, especially AstraZeneca, but also the other vaccines, and we have communicated on that. And this is where we have performed observed versus expected analysis, and we have seen that we did not have cases reported higher than the number of cases which was expected based on the background incidence rate. And the second communication was associated to the fact that we are currently also investigating the thrombotic events, not only associated to a specific batch, but more globally, since some member states decided to suspend not the vaccines, but to suspend some batches or the vaccination of AstraZeneca before having this full assessment of the thromboembolic event. So it means that the system works. We work as soon as we have some cases to make the good assessment. And finally, some information that was coming from publication of course of cases is also some delayed reaction. Quite importantly, these are not serious delayed reaction, but this need to be investigated. This was published in the New England Journal of Medicine, and it is in fact currently covered by the current patient information leaflet. What was not clearly mentioned is that we can have some cases with a median onset based on 8 days, and the majority coming after the first dose. In some cases, after the second dose, we had also these cases. But this is something, again, a signal that needs to be followed. And so I finished my presentation because I was asked to discuss these 4 vaccines that are now on the market, at least at EU level and in Belgium. But we have, as I have said, 2 other vaccines that are on the table of the European Medicine Agency for rolling review. But on top of that, we have also a new one. Of course, you have heard about the Sputnik, the Russian vaccine, for which we have publication about the efficacy in the Lancet journal, and which is under rolling review at the level of the European Medicine Agency since the 4th of March. And there are also current discussions to have at least one Chinese vaccine under rolling review at the level of the European Medicine Agency, which are inactivated virus vaccine when compared to the Sputnik vaccine. And this is the publication for the Sputnik vaccine, but again, this needs to be assessed more in detail, because at the level of the European Medicine Agency, we work and we focus not only on the clinical benefits of the vaccine and the safety, but also the quality. So all the data has to be in compliance with how we authorize drugs at EU level, quality, efficacy, safety, and the risk management plan and the post-marketing. But quite interestingly, it is also an adenovirus, prime boost adenovirus vaccine. We see high efficacy, but in hospitals that are mainly in Russian hospitals. So it means that based on the variant of interest, variant of concern, we might have also some information to be provided. And importantly, again, some efficacy quite high on moderate and severe cases. So I will not discuss the difficulty that we have today with the variant to compare, for instance, the efficacy of the vaccine. You have seen that in clinical data, depending on where the vaccine has been studied, when it has been studied, it's quite difficult to have estimates to compare in between vaccines in terms of efficacy. But what is quite important is that you see that with, for instance, the Janssen vaccine, it gives a strong efficacy for severe, moderate and severe cases, even in regions where we have some specific variant of concern. So I will not discuss these variant of concern. This will be proposed by Piet and Marco. There are some good publications that also provide not a bit, I would say, not really up-to-date estimates of the efficacy of some of the vaccines in these variants. It will be proposed. So my final slide is the whole ongoing discussion that we have about efficacy, 1 dose versus 2 doses in seropositive individuals. And you have seen that maybe New England Journal of Medicine was again published some information on that. the absence of those in seropositive individuals, as it is proposed, for instance, in some countries or proposed by the WHO and the CDC in some cases. But what do we use as correlate of protection? We do not still have validated correlate of protection. What do we do depending on the variant in this case? There are many studies ongoing as part of the risk management plan, post-authorization safety studies, but also efficacy studies in different countries, for instance, cancer immunocompromised frail patients. The variant, I will not go to that, and Marco will go into detail. The clinical trial is still ongoing. We will have additional data coming from ongoing clinical trial, long-term efficacy and safety. For the new clinical trials, there are ethical concerns on how we do new clinical trials when we see the efficacy of many of the vaccines. The real-life data, you see that it's starting to provide interesting data, both in terms of efficacy, but also in terms of safety, not only for spontaneous report, but also for a series of post-authorization safety studies, including registry. So my final slide is this one. You have seen that it is developed quite rapidly, this vaccine, but with the variants, with the safety data, the efficacy data, effectiveness data, it's not the beginning of the end of the pandemic, but more likely the end of the beginning. The vaccine being really the game changer in this case, and the different vaccine will help surely to come back to, I would say, not a normal life, but a life for which we can hope to have now good tools in terms of preventing the disease to happen, especially the hospitalization and the more severe cases. I thank you for your attention.
Thank you, Professor Dogné, for this thorough review of efficacy and safety of the vaccines available on the market. I see questions coming in, and they will follow in the discussion. It's my pleasure to introduce now Professor Piet Maes, active in the Rega Institute in Leuven. He's an expert in the ecology and evolution of viruses and strongly involved in the detection and analysis of SARS-CoV-2 variants in Belgium. Professor Maes, the floor is yours.
Thank you for this nice introduction. So I would like to take the opportunity to talk a bit about SARS-CoV-2 variants in the next 15 minutes. So I think it will not come to a surprise. I think everybody knows that viruses mutate, and also the coronaviruses, they mutate. But the big question here is if it really matters and what the possible impact is of these mutations. So if we would compare the coronavirus evolution rate, the SARS-CoV-2 evolution rate with other viruses, we see that for an RNA virus, the evolutionary rate is actually quite slow. So scientifically, it's 10 to the minus 3 substitutions per site per year. In human language, it means that more or less 2 substitutions will arise on a monthly basis. This is actually for an RNA virus with this genome setup is not a lot. And we can also see that if we look at transmission chains, that if we follow up clusters, that we see that the transmission outpaces the evolution. So that means that we see a lot of identical sequences if we investigate sequences or patient samples, which is actually a good thing. So that means that the evolution goes slow, that the mutation rate or mutations will arise on a slow pace. So that means that we will always have the time to act if needed. So before we really start, I want to come up with a few definitions. Luckily, the WHO finally gave some very important definitions on the end of February, especially to define what a variant really is, what a variant of interest is, and then what a variant of concern is. So a variant, very logic. The difference between a mutation and a variant. A variant is a collection of specific mutations that give a virus some advantages or disadvantages. And then in that same definition, a variant of interest is that the virus phenotypically changed in that way that it has some mutations that can be associated with a specific implication. So more infectivity, or for example, worse disease. And very important as well, you can detect, or on daily basis, we detect new variants. It's of course important that it's a variant, if it's a variant of interest, that it appears multiple times so that it's able to cause community transmission, or that it can cause clusters of COVID cases, or that it's detected on multiple countries, multiple sites. Now, a good example to show you that a variant is not equal to a mutation, and you probably all know about the first mutation that was detected, the D614-2G mutation in the spike. This is actually an interesting case that this mutation is thought to make the virus more infectious, but it does not appear to make the disease more severe or does not help the virus to escape vaccines or immunity in general. So this happens, the mutation, an aspartic acid mutation to a glycine, causes a loss of one hydrogen bond between the chain A loop and an alpha helix in the spike protein. That means that the protein is more flexible in the loop region, which makes specific sites. There is a specific motif that needs to be cleaved by a furin to be able to enter a cell. This specific motif comes more to the surface of the spike, and that means that it's more easily that it can more easily infect cells. Interestingly, the very first case, I will come back to that, was here in Belgium, was a wild type. But after that first case, we really gradually and quite fast saw that this mutation took over for the wild type, and by the end of June, not only in Belgium, but internationally, we saw that almost 100% of the sequences or the strains had this specific mutation. So you can see here a 3D structure of the spike protein. Important to say is that spike protein on the surface on a virion is composed of 3 spike proteins that are attached together. So each mutation that arises in the protein will be on the membrane of the virus 3 times. If you look at this specific mutation, you see that it's not in an immunogenic region or in an important region for the immune system. It's nicely in the middle on the surface. But if we look closer to the formation, you have 2 forms of the spike. You have a closed conformation, which is the wild type without the glycine mutation. But if the mutations arises, then each of these subunits of the spike will slightly change the conformation, and you see here that you get a more open conformation. So the furin cleavage site is here at the site, at the inside of the protein, and it makes that the furin can more easily get to the site where it needs to be cleaved. So I already said our first case, you all know Philippe Soubry, who came back from Wuhan beginning of February. He was on a flight with several other French and Belgian people returning back from Wuhan, and he was the only one positive. He actually never really got sick, luckily, but was quite long and very highly positive. And interestingly, the virus that he carried was almost completely identical to the wild type reference strain from Wuhan. It had only 3 mutations, and they were all synonymous, so they did not give rise to other amino acids in the proteins of the virus. That same month, 5 other cases were detected, Interestingly, all those 5 cases already carried the new mutation, so they had a G instead of the D here. In the month after that, in March, we here in Belgium sequenced 434 sequences, and 86% of these sequences already carried this new mutation. If we look at the difference here, it was between 7 and 12 mutations compared to the reference sequence, but only 3 non-synonymous mutations, so 3 mutations giving rise to a new amino acid here. Important at 2 in the polymerase and, of course, that specific spike protein. And if we look at the data for Belgium, we see that initially, but this is, especially in February, we only had 6 samples, so that's why it's so high. But then we saw that it's in March, April. March it was high, April it was lower, and then it slowly went up. And then by the time, by July, now we are at 98, 99% that have that mutation. Internationally, we see the same story or the same situation, and now this mutation is actually considered as the wild-type coronavirus mutation, so not a variant. So now, when do we talk about a variant of concern? So of course, it needs to be a variant of interest. It needs to have a specific set of mutations that have been seen in more than a few occasions. But in compared with a variant of interest, through very specific research and comparative assessment, it needs to be demonstrated that this specific variant has an increase in transmissibility or has some detrimental change in the epidemiology or disease. Of course, this includes virulence, clinical disease presentation, or a decrease of effectiveness of, for example, vaccines, of therapeutics, or even reinfections that reoccur. If you now look at all the variants, so I already said that on a daily basis we detect new variants. I think according to some papers, there are more than 7,000 variants of interest for the moment with specific mutations. So there are really a lot. But if you look at the variants of concern, luckily until now, there are only 3 or 4 if we take into account the P1 and P2 Brazilian variant. So we have the typical B117, the UK variant, which first arrived in September in the UK. Then we have the South African variant, the B1351, and the P1 from Brazil, which also has the P2, which has only 1 mutation difference with the P1. Now, how do we, or how internationally are those variants detected? So of course, you all know this very well, I suppose, with next generation sequencing. So there are actually 2 good platforms. There are probably more, but these are the 2 that are mostly used. The typical Illumina sequencing platform and Oxford Nanopore Technologies Me9 sequencing. Both have their pros and cons. I think the biggest advantage of Illumina sequencing is that you can really, depending on the machine you use, you can really run a lot of samples, so it's easily used for high throughputs. And if you use a lot of samples, you have a very low cost per base, so very cheap sequencing. The disadvantage, but this is less important for coronavirus sequencing, is that you have to work with short sequences and you can only generate a specific consensus sequence for it. The Oxford Nanopore Me9 sequencing, which is more and more used in the last years, the big advantage here is that you can use long fragments. There is actually no limit on the length of the fragments that you can sequence. You can directly sequence on RNA, so you don't need to use DNA only. And maybe the biggest advantage is that you can have real-time results. So it's a lot faster than the Illumina sequencing because you can check your data until you have enough, and you can easily stop the data when you have enough data for a specific analysis. The big advantage is you cannot really use it for high throughput, or at least not as high as with the Illumina sequencing, which makes the cost per base a little bit higher than for Illumina sequencing. And you have the problem with the error rate. So if you look at single read accuracy for the Illumina, this is a lot better than for nanopore sequencing. So that means that if you want to have the same quality of your consensus sequence, you need to sequence a lot more, or you have to generate a lot more data to come to the same significant result. Now, specific for coronaviruses, next-gen sequencing is very interesting to do to use a metagenomic approach, but of course here with the amount of samples and the urgency to sequence, this is not feasible to use a metagenomic approach. It's too expensive, too slow. So I think all people, or internationally, there is a consensus to use amplicon sequencing for this. So this is the way we do it. There is, of course, there are other approaches where you use other primers or bigger fragments. So what we did is that we chopped the complete reference genome in 196 fragments of around 400 base pairs, which you see here. We divided the primers in two pools, each of 400 base pairs, which are more or less an overlap between 40 to 50%. And from then on, it's really simple. You put in the PCR. When the PCR is done, you purify, you make your library prep, you put it on a machine. It needs to run we use a grid time. It needs to run like 6 to 7, maybe 10 hours, depending on the quality of the samples. After which you have a short bioinformatics pipeline where you put all the fragments together using mapping. Then you conduct some quality checks to make sure that your primer sequences are out, that your coverage is enough for each site, and so on. And then you have your final consensus sequence. So what we use, there is an international Wellcome Trust sponsored network, which is the Arctic network, which is really great. So they optimized all methods and also these primer sequences. And this is actually one of the most widely used methods now for the SARS-Corona sequencing. So if we have a quick look at the genome and at the differences that have been detected until now. So we are now at almost 700,000 full-length sequences internationally. In Belgium, here we are almost at 8,000 sequences. And so these two plots are the nucleotide differences and the amino acid differences noted. You see that almost every site on the virus of the genome has some changes detected. So the most important ones are, of course, the peaks that stand out. And there you see that compared to, for example, influenza, maybe it would have been nice that I've put a plot of influenza. It would be a completely different story. There you would see a lot more variability. So the thing to remember here is that actually there is a baseline with a lot of errors and mutations that are only seen once. And unfortunately, there are around 10,000 very bad sequences online from the 700,000, which makes that some of these, most of these baseline changes, I would call it, are noted. Okay, if you look at this is a timeline of all different variants. So there are several systems to define variants and to give it a name. So the two main ones are the one you see here, which is made by NextStrain and Ghent. And then you have another one, which is called pangolin. So, for example, the numbers, the B117, that's a pangolin lineage or definition of lineage, and the 19A, B, and so on are the ones used by Ghent and NextStrain. You see here that in the beginning of January, there were already 3 different strains. It has been calculated that the introduction of the virus this probably appeared probably around end of October, beginning of November, and then it makes sense looking at the evolution rate that you already have some variants, different variants at the beginning of January. It's important to say that these variants, that they really differ only in a few mutations. So there are only 4 differences between those, between 19A and 19B. So that's very important to remember, that the variability is very low. If you look at the end now, this was a situation from yesterday. You see that, for example, for Belgium, I think that the UK variant now positive cases is over 50%. But if we look internationally, we're still about 35–40%, which is luckily still not that high. But you see that it's getting higher and higher. The P1, the Brazilian variant, is about 5%, the same as the African, which is about 7 or 8%. Now, some specific details for the variants. So the first important VOC variant of concern was the UK variant, the B117. And what makes this strain so interesting or so important are the specific mutation, especially in the spike protein. So I marked them in bold, the typical, you all know it, the N501Y mutation, which is shared among the 3 VOCs. Why is this one so important? If we look at the 3D structures here, here you see the side view of the spike, here you see the top view. Remember that every spike protein on the surface of the virion is composed of 3 identical subunits, so every amino acid change you see 3 times. So the specific site, the 501 spike site, it's really in the middle of the receptor binding domain, at a very important site where antibodies can also bind. So if there is a conformation change here, it of course will have impact on the way the protein will work. And that's the reason, of course, why this mutation is so important. The same for the deletions. We have 2 deletions at the sites here, and then the P681 mutation, which is also which is more at the sites. Now the same, sorry, if you look at the geographic spread of the virus of this specific variant, it's especially the UK, but this is overrepresented. The UK is actually the country which sequenced the most, so it's really biased. I think they're now at almost 400,000 sequences, or already even higher than that, which is over for such a small country compared to the rest of the world. It's a lot, but you really see that it's especially Europe where these UK variants are found the most. A side note here is that in Europe and the States, they are the 2 regions where labs are sequencing the most. So that's probably also a wrong representation of the real coverage of that variant. If you go to the African variant, the South African variant, or the B1351, here this one shares again the same mutation on the top of the spike, the N501Y mutation, and it shares 2 other important mutations which are more linked to immune escape. And it's important to know that these mutations it also shares with the Brazilian variant. And again, if we look at the spread of this variant, it's again, of course, mostly situated in Europe. I already said it's a bit biased because of the sequencing efforts in Europe, but also in South Africa, where it originates. It depends on the week. The data, they differ a lot, but it's around 70 to 80% of all positive cases are now that specific variant in South Africa. And the last one, the Brazilian variant, again, if you look at the top of the spike, it has those specific variants The difference between the African and the Brazilian variant are the extra mutations in the polymerase and some different mutations. But for the rest, they actually act the same. From this variant, there is not really a lot known in terms of implications of infectivity or immune escape, but if you look at exactly the same mutations as the South African variant, I think we can make the same conclusions here for this variant. And if you look at the spread, luckily it's a lot less. It's especially in Brazil, of course, and in parts of France, and also in Belgium and Germany, and in a lesser part in the UK, where you can find this variant. Now, to conclude, until now, we only have 3 variants of concern. The UK variant, which is probably more infectious with roughly 50%, and then the South African and the P1 Brazilian variant, which probably has a reduced effectiveness for some of the vaccines and for the human immunity after an infection. But of course, these are not the only specific variants of concern. There will be a lot more in future, and especially if you look at the specific mutations that a lot of variants are developing. And then I'm especially talking about the mutations that arise in the receptor binding domain and in the domain where most of the antibodies have their epitopes. And it's important to know that I should have told that before, that those 3 variants and all those mutations that those 3 variants carry, that they arose independent from each other. So there is no link to any of the patients or to any of those variants. So that means that it's really not only due to the humans, or it really has a major advantage for the virus to develop these specific mutations. Of course, the more people that get infected, for the moment we have a huge population that carries viruses or that are infected. So that means that these viruses can very easily mutate, and they will keep on mutating until immunity kicks in and vaccination will take place. It's not always bad news. So we have specific mutations that are of concern that can probably make or in the future combine to new variants of concern. We not only need to look at the spike, probably also to other mutations in genes. There are a lot of mutations in the polymerase. Until now, not a lot has been done with these mutations. We probably will need to look into them more because they also will have a benefit for the virus. But to end, I want to talk about one variant that we've seen also in Belgium. Until now, we have more or less 300 cases of this specific variant in Belgium, in the UK, in France, and some cases in Portugal. Most of these people are associated with returning travelers from Congo-Brazzaville. For Belgium, that was the case until a few weeks ago, and now we are at 4 or 5% of the positives that have this variant, but most of these cases are not associated anymore with travel or with contact with returning traveler. It has some specific mutations which causes some concerns, but it also has a very important mutation in downstream protein, which is called ORF3A. Interestingly, this protein is supposed to be involved in apoptosis in human cells, but because it has this specific deletion in the protein, this protein is not functional anymore, and so probably this variant does not cause disease or only mild disease. For the metadata, or the data that we have from the patients that came here to Leuven, indeed most of them had no symptoms or only very mild symptoms. So it's not always a bad sign that there is a new, more infectious variant circulating. And with this conclusion, I want to end my talk.
Thank you, Professor Maes, for this nice and excellent overview of SARS-CoV-2 variants. And now it's my pleasure to introduce Dr. Marco Cavaleri, who has been working for many years at EMA and is presently chair of the COVID-19 EMA Pandemic Task Force. Marco, we have seen two very enthusiastic speakers before you, which makes that we are running over time. And I dare to ask you if you can bring your message in a concise manner in order for us to have some time left for the panel discussion. I'll be extremely grateful if you're successful in that. Thank you.
Thank you very much. Good afternoon to all of you. I hope you can see my slide and you can see me too and hear me. So indeed, I will try to be very brief and I will build on what Piet has been saying, but more with a focus really on the vaccines that will be coming in the future and with a focus specifically on vaccines that would target the variants. So just to tell you that, as you know, there are, as Jean-Michel showed you, there are 4 vaccines now approved in Europe to use a viral vector based on the adenovirus as a non-replicating virus, and also 2 using the messenger RNA technology. But there are other vaccines under development belonging to this same type of platform, but also different one, especially like inactivated whole virion vaccines or vaccines that are based on subunit with adjuvanting system. And all these vaccines are now struggling to go through into large efficacy studies. So we will have to see indeed what will be possible to do and what kind of evidence we will be able to gather with this vaccine in order to define the benefit risk in the prevention of COVID-19. So going straight into the issue of the variants, as you know, what is of concern with the vaccine is that particularly with certain variants, some vaccines may not be really protective. And I will start with a bit of the worst case that we got in front of us, which is the AstraZeneca vaccine. And as you know, they conducted a small study in South Africa. It was really a study of around 2,000 subjects randomized between the AstraZeneca vaccine and the placebo. And what they seen there is that there was not really efficacy emerging in terms of protection from COVID-19 of any severity from the AstraZeneca vaccine following 2 doses. And this was in conjunction with a period in which the South African variant that you just heard about was really the main variant circulated. So the vast majority of the cases recruited in these trials were belonging to that kind of variant of SARS-CoV-2. So really of concern. Of course, here we're talking about protection from COVID-19 of any severity, and essentially mild disease was what was collected in this study. So in actual fact, we still really don't know if protection from severe disease will be maintained with this vaccine, even in the face of this variant. And it will be extremely important that more data will be collected in order to get a good overview of whether at least there will be quite a good level of protection from severe disease. But what is also interesting and put out in their paper is that they look into the cross-neutralization potential of the sera from vaccinated individuals with the SHADOX1 vaccine. And what they seen both using a pseudo virus neutralization assay and a live neutralization test was that a lot of the subjects did not show any level of cross-neutralization against the South African variant. So here we are seeing that in the majority, or at least more than 50% of the subject tested here, and we're not talking about a large data set, but still it's quite significant to see that really the neutralization potential from the sera against the South African variant was really abrogated. And this is really of concern. And as you know, we don't have yet a correlative protection from any of the SARS-CoV-2 vaccine, but still this would show that when you completely lose your neutralization potential, you may get into trouble, and at least you will not be able to show really protection from disease of any severity. The situation with the messenger RNA is a bit different, and these are two studies done, one with the Moderna and the other one with the Pfizer-BioNTech. So on the left is around the Moderna vaccine, and what you see on the top left is that essentially a sera from vaccinated individual with the messenger RNA vaccine from Moderna using the ancestral strain showed really good cross-neutralization against the B117, so the Ghent variant. While if you look down at the bottom of the slide on the left, you still see that there is cross-neutralization against the South African variant, but this is reduced. So we are losing something in terms of cross-neutralization potential. And what also is interesting here that not only the 3 key mutation in the receptor binding domain have an impact on the cross-neutralization potential, but all the 10 mutation in the spike protein impact the level of cross-neutralization. Now, the point I said is that we don't have a correlate of protection, so it's very difficult to say whether this reduction in cross-neutralization really impact protection with this vaccine, and it will be extremely important to collect data in the field to really understand if there is any decrease in the level of protection with this messenger RNA vaccine. And on the right, you see some data with the South African variant with respect to the Pfizer-BioNTech vaccine. And what you see here is that similarly, there is a decrease in the level of cross-neutralization, but again, still maintain a certain amount of neutralizing antibodies, and therefore is extremely difficult at this stage to really say whether or what will be the impact on the actual level of protection from this mRNA vaccine against the South African variant. Still, it's notable to see that there are quite large amounts of neutralizing antibodies, so we may wish or hope that still a certain good level of protection will be achieved, even if, frankly, we don't know that yet. A few words on the vaccine that EMA gave a positive opinion today is the Johnson & Johnson Adeno 26 vaccine. And as you know, this has been studied in a large randomized clinical trials across different regions in the world, including also Brazil and South Africa. And in particular in South Africa, the studies was conducted when there was a really high circulation of the South African variant. So those data are quite relevant. The data from Brazil are also interesting, but essentially were conducted in an area where the variant that was circulating was the so-called the P2 variant, which has much less or a smaller number of mutation in the spike protein than the P1. But nevertheless, what is remarkable here is to see that despite the level of protection is somehow decreased, still there is protection, in particular with respect to severe critical COVID-19, the efficacy is still above 80%. Particularly at day 28, because what probably is interesting to see here is that it looks like protection in the face of the South African variant. It takes a little bit longer after vaccination really to mount to a full level of protection. So it looks like you need a bit more maturation of the adaptive humoral response before you can really achieve pretty much the same level of protection that you have seen with the ancestral strain, as seen like in the United States. But these data overall are quite reassuring and encouraging on the fact that this vaccine will be able to confer protection against the South African variant and possibly also all the variants circulated in Brazil. Another study that was conducted in regions where variants are circulating are the two studies done by Novavax. Novavax indeed has a subunit vaccine with an adjuvanting system made of matrix 1. And what they saw is that in the UK, with high circulation of the Ghent variant, protection was really high, about 90%. In South Africa, this protection was cut down to 60% roughly, or even less. This indeed when the South African variant was really circulating. So it's an important factor to keep in mind. But also quite worrisome in these studies was that in the placebo arm, there was a high level of seropositive at baseline, around 30%. And what they seen is that the attack rate in the seropositive was exactly the same as in the seronegative. showing that previous natural exposure to the ancestral strain would not give protection against the South African variant. So this is something that, of course, we will need more data, but is a bit worrisome in terms of what natural exposure to the one strain would confirm as protection against the South African variant. And another very interesting study that has just emerged from South Africa is this one. Again, we will need more data to confirm this data, but what is showing is that essentially subjects that were exposed to the one strain showed a reduced level of cross-neutralization against the V2 variant, while subjects that were naturally exposed to the V2 variant maintain a better level of cross-neutralization against the ancestral strain, which was something that, you know, we were not really expecting, and it will be very interesting and important to have more data to really confirm if this is the case or not. And it will be very interesting once we will have vaccines emerging with any of the variant to see what is the level of cross-neutralization against the ancestral strain as well. I will not spend too much time on this, but I think this is a very interesting paper that shows something that is really emerging, which is that essentially, if you were naturally exposed to SARS-CoV-2, probably one dose of vaccine will be sufficient. At least these studies was done with the messenger RNA and really showed that you are not really you're not gaining anything by giving a second dose. But also importantly was they showing that we should not just look at neutralizing antibodies, but look into the amount of memory B cells. And also the amount of memory B cells is also an important factor and predictor of then the booster effect once you receive one dose of the messenger RNA. So again, something that we will have to be looking on in the future once people will be highly exposed to SARS-CoV-2 or to vaccines against SARS-CoV-2, and to think about booster strategies. Now, very rapidly moving into what the EMA has been doing in terms of thinking how to approve future vaccines that will incorporate any of the new variants. And we started discussing with our international partners, essentially other regulatory agencies like the FDA and Health Canada, but also with WHO. And also we had a workshop with the International Coalition of Medicine Regulatory Authorities, which it's a group of agencies from all the continents in the world. So it's really an important group in which to agree the way forward, and we achieved that. So we were able to agree on the key elements for vaccine development for variant vaccines. And after that, we issued an EMA reflection paper in the 25th of February that really summarized our thinking about how to rapidly develop new vaccines that are based on the emerging variants. And of course, we started already talking to developers of the vaccine that are already approved in order to see how to move on in the design of the clinical study and what other information on manufacturing will be needed. So first of all, it's important for us to stress there might be a clear-cut justification for the relevance of the variant that is inserted in such vaccine, and should be based on the current need in the European Union, based on the surveillance of what is actually circulating in Europe, or is expected to be circulating in Europe in the near future, and a very good characterization of those strains. And then if this is convincing, we think also that would be important to have a global forum like WHO in order to make a clear-cut recommendation on which strain should be included in this variant vaccine. As you know, this is already done for influenza vaccine, and maybe there will be a need to establish something similar for SARS-CoV-2 as time goes by. So in a nutshell, in terms of clinical development, what we are going to expect is that small clinical trials will be conducted in order to provide some immunobridging results that would allow us to infer the protection of the variant vaccine based on comparison of the immunogenicity against the vaccine, the original vaccine, the parent vaccine that demonstrated efficacy in the field with the one strain. And this is in two settings, essentially. First of all, after primary vaccination with the variant vaccine, so essentially comparison of a primary series in naive individual of the parent vaccine and naive individual with the variant vaccine, and then to compare the immunogenicity in terms of titers of neutralizing antibodies and seroconversion according to pre-specified hypotheses based on non-inferiority on these two outcomes. And then not only in the setting of primary vaccination, but also in the setting of booster vaccination, as indeed this will become more and more common in the future. And indeed here the point would be to study what happens after a single dose booster with the variant vaccine for individuals that receive a primary series with the parent vaccine, and maybe possibly comparing to an homologous boosting, so boosting with a single dose of the parent vaccine, and to compare the immunogenicity in a similar fashion as done after a primary series. And all these elements are well described into our reflection paper, and we assume that this would allow to come up very rapidly with an authorization of this variant vaccine in order to have them timely available. Then very few words about other vaccines. As I said at the beginning, there are a number of vaccines that are still in early phases. They have not yet even started phase 3 trials and might have complications in starting phase 3 trial using a design that has been used so far, which is a randomized control trial using a placebo arm. This might not be possible, at least in some part of the world like the European Union, as vaccination is progressing and vaccine got rolled out. So at least for high-risk group, it will be a problem really to do this study and will not be considered feasible. At the same time, running large non-inferiority studies with a clinical efficacy endpoint might also be problematic, as indeed these vaccines are really effective. We don't know what is going to be the attack rate once vaccination campaigns have been rolled out to an extended fashion, and therefore also this option might not be the way to go. So what we may be left is to design a non-inferiority study looking at comparison of the immune response and specifically looking into the titers of neutralizing antibodies with respect to a vaccine that showed clinical efficacy in the field. But the problem is that, as I said, we don't have yet a course of protection, and we don't even know to what extent you can make this immunobridging to vaccines that have different platform and therefore may use different mechanism of protection. Therefore, we need to discuss this in depth to really understand what would be the best way forward. But we are really open to discuss this option, as we are really struggling to see what else can be done. So this is just my summary slide. It just said what we've been doing at the EMA and what is coming next are the vaccines for CureVac, Novavax, potentially also the Sputnik vaccine from Gamaleya that all started rolling review. Also to say that we are monitoring quite closely the real-world evidence, as Jean-Michel was saying, and we are ready to take on board whatever important evidence may be coming from this study. As said, the guidance on the variants from the EMA is out, and we are ready to discuss with developers how to really move forward into approving these vaccines. And as well, we will be discussing with developers a way of really determining what is the benefit for second and third generation vaccines in the context of protection from COVID-19. And of course, we really urge the entire scientific community to go a bit faster in having a good understanding of course of protection, or at least a minimum understanding of what is the role of neutralizing antibodies versus T-cell responses, and as this will help a lot regulators into navigating the future approval of vaccines for COVID-19. So I think I will stop it here. Thank you very much.
Thank you very much, Dr. Cavaleri, for your very interesting talk on variant vaccines and sharing your insights on what we may expect in the near future. With this, I would like to open the Q&A session. We're a bit, behind schedule, but we would like to spend about 20 minutes asking questions or answering your questions. And I would also like to welcome two panel members who will join us, Dr. Johan van Hoof Janssen from Janssen and Dr. Stefan Lockhart from Pfizer. Welcome and thank you very much for joining us. I will start with the first question, and because we have, many questions and we would like to answer as many as possible, we appreciate it if you can keep your answer brief indeed, but of course sufficiently clear. I would like to start with a question for Dr. Van Hoof, Johan Van Hoof. Just gonna look on the screen. Apologies. Can some people already have pre-existing immunity against the vector in the Janssen vaccine or other vector vaccines? And could that result in failure to build immunity against COVID? And maybe it's a question that I would like to add. Is it necessary or useful to measure antibodies after vaccination?
Thank you for the question. It's a question that comes up very often, and it's a very valid question. Fortunately, we have quite some experience with this vector in our R&D programs. And so by the time that we started the COVID program, we already had experience in more than 120,000 people, of which a large cohort has been vaccinated in Africa, where you do see a lot of pre-existing immunity against H26. To make a long story short, we don't see interference between that pre-vaccination immunity and vaccine take. It is probably related to the fact that the titers that you see as pre-vaccination immunity are relatively low compared to adeno5, and that the viral load that you have in your vaccine is large enough to compensate for that eventual interference. So it's a good question that I'm happy to say that we know and have data that show that this is not the case. We have again seen it now in South Africa, where we also have seen no interference in vaccine response.
Thank you, Dr. Van Hoof. A question that came in from one of the attendees is, a question expressing some concern about the whole approach of vaccination at a global level, what we are doing today, using as specific vaccines or as antigen as the target. And the person asking the question is wondering whether this approach is not more dangerous than beneficial by putting a lot of pressure on the virus and moving into selection of variants. So the key point of the person asking the question is, are we not making things worse by mass vaccination with S-only vaccines rather, and will we not prolong the pandemic by this approach? I don't know who can answer, but possibly Professor Maes is well placed to address this question.
Yeah, I can start with that. So it's a valid question, but history never showed that this happened before. I think that a very good example are the Ebola outbreaks. So especially the last ones in North Kivu, where there were vaccines tested and used in the population. And it was quite effective. And if we look at the difference between SARS coronavirus evolution and Ebola virus evolution, Ebola viruses, they have a much faster evolution rate. So between each patients, there are really sometimes very huge differences. There are always important changes, and still it never caused a problem that there were vaccine escape mutants there. So I think the biggest thing here is time. There is simply no other way of using vaccines. I think developing other vaccines will take too much time and will take us several years. I think that the vaccine approach who is here now is a good one. It's been used before, and even if it will not. I don't see the risk there.
So,
Is it okay? Okay, thank you very much. The next question is on, seeing the intrinsic biologic variation in clinical trials with the different vaccines and the not standardized setup of these trials. do these numbers we have seen today and have been presented, like 66%, 80, 90, according to you, and maybe Professor Dogné could answer that, or Dr. Cavaleri, or whoever would like to answer this question. Can these differences in efficacy numbers can be considered as really different or not? Or would we need another type of study to assess that?
Yes, I may maybe start, and then Marco can, of course. First, I would also react to the previous question. I think that the main mutation that we have seen and the variant of concern that we have seen, the British one, the South African and the Brazilian developed before the vaccination campaign. So it just means that clearly without vaccination campaign, you have already vaccine of interest, a variant of interest that developed. So I think that the vaccination itself will not have an impact, a huge impact of creating new variants, but will help maybe to limit what we need to have as limitation, hospitalization and cases. So that was my contribution to the previous question. To this question, of course, what I have said is that the endpoints, if you compare in the different clinical trials, may be somewhat different. And with, for instance, for the Janssen vaccines and for the mRNA vaccine, the endpoints were somewhat maybe a bit different. And what I have said is that indeed we do not have head-to-head comparison of the vaccine at the same time in the same region, in the same clinical trial over 30,000. And I suspect that it will not be the case in the future. So what we need indeed is not to compare to the estimates of the different vaccines in the different clinical trials. And at the level of the EMA and the FDA, it was understood that the benefit risk of the vaccine were not a comparisons in between the vaccine, but an efficacy of the vaccine in the clinical trials that need to be over 50%, I would say. And so I urge usually people not to compare vaccine, for instance, believing that AstraZeneca is a worse vaccine than, for instance, Pfizer vaccine. And we can compare every estimate that you want, but when they have been studied differently in different population at different time, basically even on different variants, it's quite difficult. So now we need to focus on efficacy data coming from clinical trial, take these data as they are, and focus on effectiveness data. And I see that effectiveness data for different vaccines may be a bit different when comparing to the clinical trial when they were studied. So this is it for me. I don't know if Marco wants to complement.
No, I think Jean-Michel covered all the main points. Just also to say that also the attack rate was not the same in the different trials conducted in different regions, different point of time. So we have to be extremely careful in never over-interpreting such data and making direct comparisons.
Thank you. Another question we receive is, can we expect better protection with inactivated vaccines versus the variants than with the existing ones? Possibly assuming that by introducing inactivated vaccines, you confront the body with a larger array of antigens. But of course, you can speculate, but I guess it's difficult for all of us to think what the impact can be on final effectiveness. But if anyone feels the urge to add or to speculate, feel free.
I guess indeed, it's just a speculation. Of course, we don't really know. And to be honest with you, we are still waiting to see exactly what the data are from all these inactivated vaccines and how much effective they are. Yes, of course, with inactivated, you have other antigens. So like the N protein that we know is a protein that elicits an immune response, could have a role. but we also know that in terms of humoral response is not immunodominant, and the spikes is really driving the majority of the impact in terms of antibodies response. So it's very difficult to say it also in terms of T-cell responses. We could argue that maybe there could be some advantages, but it's very difficult to say that today.
Yes, and I may add also that up to now the data that we have are quite scarce and quite limited, and hopefully we will have inactivated vaccine on the table of the European Medicine Agency that will be assessed more in details, that when we have with now some information coming from China from the study. But what I can say is that we can speculate how much we want. What is, I would say, the gold standard is the data from the clinical trials. And when you have clinical trials with efficacy over the 90-95% on hospitalization or on severe cases, I think this is the proof of concept of the vaccine, for which we did not eventually expected such a high efficacy. So I think that believing that with inactivated vaccine we will have higher than 95%, hopefully it may be, but I do not think that we will reach 95%. So I think that we should not speculate on the platform, but see, as we have just said, the data from clinical trial from the different vaccine. Hopefully, I think that the target, the spike protein, is the good one, since the different platforms targeting the spike protein leads to tremendous results, whatever mRNA vaccine or adenovirus vaccine. It seems that the cellular immunity associated with adenovirus is maybe more interesting than humoral responses, and we have seen that, for instance, with some data with adenovirus. So I think, of course, they are different, but the data from the clinical trials are the one that counts in terms of efficacy, and the effectiveness study that I have presented, for instance, for mRNA vaccine in some countries are also the proof of concept in real life. Of course, the duration of the protection, the impact on the duration of the protection, the impact on the variants need to be understood, need to be studied in real life. The effectiveness needs to be studied. But I think that who in the room would have expected that with, for instance, the mRNA vaccine, we would have 95% efficacy? Not me, in fact.
Thank you very much. Today it was on the national news in Belgium. The Prime Minister actually communicated that children will also be vaccinated. Some people were surprised because children, at least in Belgium, had not been mentioned before. We know that companies are planning their clinical trials or are already conducting the trials. So I would like to ask Dr. Van Hoof, maybe if you could comment on the plans Janssen has with regard to clinical development of vaccines of your vaccine in children and adolescents.
Thank you. Indeed, there is increased interest in vaccinating the pediatric population. I see some regional differences. In the US, there is an extreme high interest in accelerating that. They really want to vaccinate all children by September 2021 to allow to open the schools again. I think in Europe, there certainly is also high interest in this. The question here is how fast you want to go in that vaccination. Traditionally, you want to start vaccinating pediatric population only once you have your efficacy signal to make sure that you really expose these children to a vaccine which has demonstrated efficacy. That's the point where we are now. We have started pediatric studies both in the US and in Europe. We start with pediatrics in the US goes up till 18 years of age. In Europe, it's 16. So we are, for the moment, vaccinating the 16 to 18 years or 17 years already in the US. Hope to have very rapidly the data, safety data on those. And then we are going down in ages. And for the moment, we plan to go down to school entry, 3 years of age. But afterwards, we also want to go into the children. We are in contact with the European Agency and with FDA, and it has been that pediatric plan, so-called PIP, has been indeed part of our submission to the agency. So pediatrics is important, and we would like to go as fast as we can, but of course we want to make sure that we respect the safety of the children we vaccinate. We know that these vaccines are somewhat reactogenic. And that's one of the reasons why we want to make sure that we go with the right speed, having sentinel subjects to vaccinate first and eventually going with lower doses.
Thank you, Dr. Van Hoof. We have a question for Dr. Lockhart. We can't see him, but he is online. And the question relates to the preservation, the storage of the vaccines, mRNA vaccines in particular, knowing that the Pfizer vaccines require ultra-low temperature. Can you explain why this is needed and if there is any perspective to have a less need for ultra-low temperatures and an easier way to handle the vaccine?
Yeah, thank you. I'm sorry you can't see me. It looks to me as though my camera's on, but never mind. Yes, it is an important issue, and clearly we took some key decisions early on because in the pandemic situation, we wanted something that would be rapidly available. And in the pandemic situation also, we think that having a formulation that can be used, particularly in large centres where large numbers of people can accumulate for their vaccination, is feasible, and it will be less feasible as we move on. So we are developing formulations that will be more thermostable and easier to store and transport as a matter of some urgency, but it will take a while to get those available.
Very much. We had a lot of questions on the impact on fertility and whether we are sure that these new vaccines do not impact fertility. I don't know among panel members who would like to take this question to reassure the public, our audience.
Well, allow me, perhaps. I'm a clinical scientist and not a molecular biologist, but we take these such risks very seriously, of course. And we have performed a reproductive toxicology study, as indeed all the manufacturers will have performed it, to look very carefully at whether or not there is any risk before or during pregnancy. And that has been completely clean. And it's something that the regulators absolutely required. Many people asked us why we didn't go earlier into studying the use of the vaccine in pregnant women. And the answer was because we didn't have the reproductive toxicology study results available. They take many months. We started them practically as soon as we developing the vaccine, but still it took us until around November, December last year before we had that data available, and we wouldn't have gone into pregnant women without that. So the other fact from our perspective is that RNA is simply not incorporated into the genome, and therefore we don't understand why anyone thinks that there's a theoretical risk of that incorporation into DNA and germline damage. There's no evidence for that.
Thank you, Dr. Lockhart. I don't know whether Dr. Van Hoof wishes to add.
As you know, we are using the adeno 26 vector, for which we have done indeed also historically these reproductive tox studies for other antigens. We are vaccinating pregnant women in DRC as part of the mass vaccination program in the Kivu area and soon starting up also in other parts of DRC. We have a large-scale vaccination program running in Rwanda, and we have a study running in pregnant women where we specifically look also at the outcome of children just out of precaution. Based on the data we have, we also did the toxicology study for specifically with the COVID insert. And so based on all of that, we do feel comfortable that there is no risk related to this. I think it's also indicated in the package leaflet now that depending on the risk-benefit ratio, that vaccination could be considered. This being said, we also are starting registration and pregnancy registry to make sure that we document also the pregnancies that occur during the vaccination, and we want to follow up on the children. But so I do feel that there is quite some scientific evidence now that actually documents the safety of this product also in case of pregnancy.
Thank you, Dr. Van Hooven. Then I have a final question, considering the time. A rather general question that also came up is whether the new platforms, mRNA, adenoviral vectors, will be stayers, will remain and become common good in vaccinology, or whether they will be related or stay within the COVID area, or do you expect these new vaccine platforms to be more widely used in the near or far future? A question for probably several of you.
But I can say, as I already mentioned, for us it is one of the key pillars of our strategy. We do have in the clinic several vaccines, RSV vaccine, where we soon are going to share data that we obtained in a clinical proof of concept study. We do have an HIV vaccine study running in South Africa, where we have the first readout results summer this year. So we certainly believe that adenovectors can be an important player, an important tool in new vaccines. Of course, we realize that you better have several tools in your toolbox to make sure that you optimize your vaccine design.
I can say something similar. I mean, I think the mRNA technology has actually been bubbling under for many years. There's been a lot of work going on to develop it to the point it's at now, and it didn't really appear overnight. It wasn't that the pandemic started and our colleagues at BioNTech said, let's make an mRNA, haven't thought about it before, but let's make an mRNA vaccine for this particular antigen. They'd actually been developing this technology for over a decade. And interestingly, their primary interest was not infectious disease. They were developing it for oncology, immuno-oncology use. And that gave them a great deal of experience, and possibly also in a population where there's a slightly different assessment of benefit risk. But now, obviously, we've been into very large numbers of the general population, and frankly, no particular safety issues have arisen. I hesitate to say none. Obviously, there's been questions like anaphylaxis in a small number of cases, and other things will no doubt appear. But broadly speaking, it's proved to be a platform that's very appropriate for large-scale infectious disease immunization. And I can assure you we're not the only company that have thought about that. So I think it'll be around as a platform from many groups.
Okay, thank you very much. And with this, I would like to, wrap up this webinar. We have reached the end, and thank you very much for staying with us at this late time. I would like to thank everybody who contributed to the webinar. Thank first of all the three speakers for sharing their insights, their very inspiring talks. Thank you as well for our two additional panel members for joining us and answering the many questions we received. I know all of you have a very and extremely busy agenda, so we appreciate this enormously. We would like to thank our strategic partners, of course. I would like to thank Fran van Heuverswijn, who organized and coordinated this webinar. And last but not least, of course, we would like to thank you for your interest, for joining us. We really welcome and appreciate your feedback, so please feel free to complete the form. This enables us to refine or ameliorate the next editions of our webinars and the other events we will be organizing. And again, for the physicians, if you want to request for accreditation, please click on the Q&A symbol in the upper right corner. You will find a link, and that will give you access until midnight to complete your name and RISIF or INAMI number. Voilà. With this, I would like to end and thank you very much and stay safe. Bye-bye.